Analytic Epidemiology, Overview of Study Design, Part 5, the last part of this particular series of lectures. In this lecture, we'll review and summarize our discussion of the different kinds of observational study designs that we have covered in the previous lectures. And then we will talk a little bit about some potential sources of bias and error. This slide shows the application of different observational study designs. So we're going to look at ecological, cross-sectional, case control, and cohort studies, and particular objectives for which each kind of study design might be appropriate. The first objective we might have would be to look at rare diseases. And for that, both ecological and case control studies work well in that we can identify groups where rare diseases occur and then look for their particular exposures that might have happened. For looking at rare causes of disease, cohort study works the best in that we can identify groups with particular rare exposures and then follow them over time to look at the development of disease in those groups with and without these rare exposures. If we're interested in looking at a particular exposure but looking at multiple effects of that exposure, again, cohort study is the best approach for that, in that we can, at the beginning, look at a given exposure, ensure that the group is exposed to it, at least the exposed cohort, and then follow it over time and just look at various endpoints. If we're interested in looking at multiple exposures of disease, other causes of disease, the case control study design is the best in that we can look at a group of people with a given outcome and then go back and ask about or look at multiple exposures. For looking at the time relationship between an exposure and an outcome, cohort design is the best in that we're able to, particularly with a prospective cohort study, determine exposure at the beginning, make sure that we know that well, and then follow people over time so that we know for sure that the exposure precedes the development of any disease. In terms of a direct measure of the incidence of disease in a population, again, cohort is best for that. We can actually follow people over time and determine whether or not a disease actually develops and count them. And finally, if we have a long latent period for a disease, case control studies work well because they can be done more efficiently, both in terms of time and cost, compared to, for example, cohort studies. Although cohort studies could be used, but sometimes the cost and time length are prohibitive for doing those. This slide shows some of the advantages and disadvantages of different observational study designs. We will, in fact, be discussing most of these kinds of factors here later, in particular selection, recall bias, and confounding. We have discussed in our lectures today issues related to loss to follow-up, time, and cost. And as we have discussed in loss to follow-up, that can be a problem in particular with cohort studies, but less so with case control studies. In terms of the time required, cohort studies, again, take a long time to perform, and ecological, cross-sectional, and case control studies require less time. In terms of cost, again, cohort studies are the most expensive, ecological studies least expensive, with cross-sectional and case control studies in between. Again, we have these potential problems with the study types, but We have to determine just exactly what we really want to do and what's the purpose of our study and our objectives, and to determine exactly what study type we should use. As I mentioned, there are potential errors in epidemiologic studies. We will be studying this in a later class. Sources of error can either be random or systematic. Random error is when our sampling or our measurement diverge from the true value by chance alone. And those can be due to individual biological variation and sampling error, or measurement errors, which again happen to be random. And the best way to deal with this kind of error is typically a larger sample size and careful attention made to the measurement in the study. Systematic errors can be caused typically by problems with the selection of people and their allocation into certain groups, either exposed or unexposed in the case of a cohort study, or, for example, into randomized controlled trials, or a bias in terms of the selection of cases in controls. There also can be classification or measurement biases where we actually have difficulties with determining whether or not, for example, a person is exposed or not exposed to a given factor.